Assalamualaikum warahmatullahi wabarakatuh. Greetings to both of our examiners, Dr. Shida and Dr. Shazni. I'm Saidatul Sofea from SMPKH5 with metric number 049433. Today I will be presenting my final year project 1. My title for the project is The Effect of Pasture Intake on Prevalence of GIT Parasites at Different Ages of Capra aegagrus hircus Female Boer in Ubay Farm. My supervisor is Dr. Nur Atika Mat Yusof. In my background study, I found out that ruminants animals are very susceptible to GIT parasites. Small ruminants such as goats and sheep may have many gastrointestinal parasites. According to Margot 2006, animals that are grown by pasture-based system of feeding are absolutely susceptible to the exposure of gastrointestinal parasites. Tropical climate in Malaysia, which is moist and warm, are perfect for the worm growth. Therefore, it can harm the animal if the pasture is not properly managed and observed. Parasites are very dangerous as it fed off the host. It may cause anemia and make the animal to go waste and even death. Goats are more susceptible to GIT parasites. Unlike sheep develop higher and stronger level of immunity around 12 months old compared to goats which obtain lower level of immunity. Examples of parasite species that accumulate in GIT are Haemonchus contortus, Emeria species, Teladosargia circumcincta, Trichostrongylus species, and many others, according to Powell et al. 2004. GIT parasite can cause a huge loss in goat production where it may cause disease such as anemia and even can cause death. Pasture contamination is one of the factors for accumulation of parasite in animals. Pasture contamination is caused by feces of animal during feeding. Feces that is accumulated by parasites can contaminate pastures and then ingested by other goats. Parasites larvae can survive on pasture at stage of L3, and it can still be protected when ingested. Pasture contamination can also increase the population of antihelminthic resistant parasites and can cause it to be harder to get rid of. Significance of studies. Studies of internal parasitism are very crucial as they are needed especially in the management and farm planning. If diseases are overlooked, it absolutely can cause loss to farmers and producers. According to Kaplan 2004 and Zajac and Gibson 2004, findings of parasites are important to avoid the development of antihelminthic resistant worms. If the issue is uncontrollable, it can cause more losses in future. The objective for my project is to determine the effect of pasture intake on the prevalence of gastrointestinal parasites in female Boer goats in Ubay Farm. Next is to compare the prevalence of GIT parasites between different ages of female Boer goat in Ubay Farm. Lastly is to identify the types of parasite between different ages of female Boer goat in Ubay Farm. This is my literature review. Materials and method. The project will run in Ubay Farm, which is the largest Boer goat breeding farms on Malaysia East Coast, which have more than 1,000 livestock. These imported breeds from Australia consist of full-bloods and local Boer, which is originated from Australia. The location is in Kampung Telaga Papan, Bandar Permaisuri, Terengganu. The animal selection are female Boer goats, Capra aegagrus hircus. The animal will be randomly selected following different age categories, which are 5 to 12 months old, 1 to 3 years old, and 4 to 7 years old. For each category, 7 female goats will be randomly selected. Next is pasture feeding. According to Ubay Farm, 2 types of pasture that is used for animal feeding over there, which are humidicola and Napier. They are also fed with pellets. Each group of animals are fed different amounts of pasture, such as doe are fed with pellets in the morning for 450 grams, pasture for 1.2 kilogram in noon and 2 kilo at evening. Meanwhile, kid are fed ad libitum in morning, noon and evening. The process of project goes with humidicola or napier will be fed to the goats, and then we will run the FEC, fecal egg norm.
The materials needed for the experiment.
Identification of parasites will use three methods, which are modified McMaster technique, flotation technique, and sedimentation. Both McMaster and flotation technique uses the same flotation solution, meanwhile sedimentation only uses tap water. For McMaster and flotation technique, the fecal are firstly collected using proper technique to avoid injuries to animal. Fecal is then kept in cold storage for the next step. Fecal is weighed for 2 grams and then crushed in mortar with 28 milliliter of salt solution and then wait for 5 minutes for it to soak well. Then use a fabric strainer to sieve the mixture into the cup. Press the feces using spatula or tongue strainer. This solution can be used for both McMaster and flotation technique. For McMaster method, immediately take dropper and drop the strained solution into both McMaster chamber. Insert carefully to avoid bubbles. Fill the entire area and then let the slide rest for 5 minutes to allow eggs to float to the surface. After 5 minutes, immediately place under microscope and observe and count the eggs. The data is then collected.
This is the flotation technique. Next is the sedimentation technique.
Identification of parasites. These are the example of parasites that can appear, which are Monasia species, Coccidia, and Emeria species. Statistical analysis. The data that will be collected after the project will be analyzed using single factor one way ANOVA. It is also will be presented plus minus SD as well as significance of P less than 0.05. The statistical analysis will be performed in Minitab 17 analysis software. My expected outcome, parasitism in animal are expected to become increased as the amount of feed increase. Identification of parasites may be showing a lot of Haemonchus and Coccidia parasite as there was once outbreaks of the disease in Ube farm. These are the references used in my project. These are the Gantt chart for my FYP flow. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.